Protein synthesis occurs in two steps, transcription and translation. Transcription is how mRNA is made from DNA, and this process occurs in the nucleus. And transcription will be broken down into four main steps. The first is when an enzyme, RNA polymerase, unzips the DNA. That way, RNA polymerase can come in and add RNA nucleotides matching up nitrogen bases. Adenine will go to uracil because there's no thymine in RNA, and the guanine in DNA will always bond to the cytosine. Once that process hits a termination signal or a stop codon, the RNA polymerase will release the new single-stranded mRNA strand. That mRNA strand is then free to travel to the ribosome, and the DNA will then come back together. The second step of protein synthesis is translation, and this can be thought of as changing the language from DNA to a protein, which is responsible for all of our traits. With mRNA, there are 3 nitrogen bases that are read at a time. Those 3 nitrogen bases are called codons, and each codon will code for 1 amino acid. There are 20 amino acids in all living things, and their unique sequence of amino acid is what determines the traits of individuals. So the same 20 amino acids are used in all living things, but what makes them different is the order of those amino acids. With translation, we have a common start codon that will initiate this process, and that is AUG, which is translated to the amino acid methionine. Translation will occur in 6 steps. The first is that mRNA and the ribosome will bind together. The tRNA anticodons will bond to the mRNA codon. And that's important because the tRNA also carries the amino acid. Those new amino acids bond together in a chain, and the tRNA will release that amino acid as that amino acid joins the chain. The ribosome will continue to move along the mRNA strand until that termination signal or stop codon is reached. And then the new protein or the polypeptide is released, and everything will disassemble and pull apart. With protein synthesis, mutations can occur, and these are any mistakes that occur in the DNA during replication or transcription. Most of the time we have proofreading enzymes to make sure that these mistakes don't happen, but these enzymes can miss something. There are 2 types of mutations, a point mutation and a frameshift mutation. Point mutation is when one nitrogen base is either added or replaced with another. And this can change one amino acid. And an example of a type of mutation that has undergone a point mutation is sickle cell anemia. A frameshift mutation is when one base is added or deleted. So that can change all the subsequent amino acids. Most of the time, frameshift mutations can cause more harmful effects. And some examples of traits that are caused by a frameshift mutation would be Huntington's disease, Tay-Sachs disease, and albinism.